Hi, my name is Laisa, and today Lachlan and I are going to talk about getting more wheat Sawfly parasitoids on the landscape. The insect on the left is the wheat Sawfly, and the orange one on the right is a female parasitoid. The species name is Brachycephaly. These parasitoids suppress wheat Sawfly populations.
This agricultural landscape is typical of much of Montana wheat production. However, wheat is relatively new on this landscape with none grown here before the late 1800s. Before that, imagine a mixed shortgrass prairie consisted of communities of grasses, forbs, and shrubs. There were also native insects present on the prairie, including what is now called the wheat-stem sawfly, and also parasitic species that coexisted with these. Nowadays, Montana is a major producer of both winter and spring wheat. And since the first record of damage in 1916 near Bainville, the wheat sawfly has become a very serious pest in Montana wheat.
Looking at this damage, there are two things that are evident. First, much of the wheat is lodged due to stem cutting by wheat sawfly larvae. Secondly, the wheat heads are not as robust as they should be, because seed weight is reduced about 20% compared to stems without the larval feeding. These losses amount to $40 to $80 million annually in Montana. Overall losses in North America are $350 million annually. No single method has effectively reduced wheat sawfly population, including rare chemical control and cultural practices with a major emphasis on host plant resistance. But there is also ongoing biological control by parasitoids, which is what we are going to focus on today. First, what is a parasitoid? Parasitoids lay their eggs on or in the body of other arthropods, usually other insects. The larva from this egg is going to develop by feeding on the host, in this case the wheat sawfly, causing its eventual death, and thus preventing more losses that could be caused by the wheat sawfly. When a stem is cut by a wheat sawfly, there is a loss of 10 to 30%, which is completely eliminated by parasitoids. In fact, one way to think of this is to consider that a stem containing a parasitoid is a good one that is going to yield well. The wheat sawfly parasitoids are Brachycephaly and Brachylysogaster. They are the only two species of parasitoids known for parasitize this pest in wheat stems. In this picture you can see the wheat sawfly larva inside the wheat stem and the parasitoid feeding on it. It's important to notice that only the immature parasitoid is feeding on the host, but not the adults. This leads to one of my questions of interest. How could we increase the success of parasitoids in parasitizing wheat sawfly? To start answering this question, I've been conducting experiments at the lab with parasitoid nutrition. For that, I use artificial diets containing sugars, and I also have water as a control. Then I analyze for how long these female parasitoids are living. In these graphs, we have the different diets on the x-axis, and on the y-axis, the number of days these females were alive. When we compare water to the other diets, it's noticeable that diets containing sugar are increasing these parasitoids' lifespan. By increasing their lifespan, they are going to be able to parasitize and kill more wheat sawfly. These results show us the importance of having food resources such as nectar or extrafloral nectar available for these females to feed on under field conditions.
Because we know nectar is an important food source to parasitoids, and flowering species often produce accessible nectar, we are interested in how these resources naturally occur on Montana's landscape. To address this, we have been sampling a relict prairie site for the presence of flowering plants. Our study area is located at the Northern Agricultural Research Center south of Havre. Throughout the growing season, we make 6 trips to our 30-acre study area. The purpose of these trips is to map the distributions of all flowering species found within the site. To do so, we mark out 10-meter transects, spaced 10 meters apart, and record the locations of the flowering plants present. So far in our fieldwork, we have recorded the locations of just under 30,000 plants, belonging to 53 different species. The most abundant species are white prairie aster, yellow sweet clover, sagebrush, hairy false golden aster, and yarrow. Based on the literature, we know some of the native flowering plants observed in our site have the potential to be used as a nutritional source for parasitoids. Those species are yarrow, hairy false golden aster, velvety goldenrod, and alfalfa, which also makes for a good cover crop. The data we have collected offers insights as to how we can integrate these species across Montana's agricultural landscape. This project has also presented a unique opportunity to gain knowledge as to how wildland flowering plants occupy undisturbed areas. The dataset we have compiled offers insights as to which species can be reintegrated to benefit both parasitoid and pollinator populations. We would like to say thank you to all the folks at the Northern Agricultural Research Center, as well as all the organizations that have lended us financial support, and especially to the professors at MSU that have lended us guidance and help throughout this project. Thanks for taking the time to watch.